Hello, everyone. My name is Matthias Palmer. I am a third year here at New York Medical College. And today I'm back to go over basic immunology. Today we're going to do kind of a comprehensive review of the basic immunology in hopes of solidifying some concepts before you begin clinical immunology here over the next couple days and weeks. So some objectives for today. We're going to review the concepts of basic immunology and hopefully provide a framework for understanding immunology as a system instead of separate individual compartments. So our plan for today, we're going to go through innate immune system responses, talking about both the innate and the induced immune response. We're then going to discuss the adaptive immune system, talking about T cells and B cells. And then finally, we're going to put it together in the entire immune response by talking about exposure to pathogens as well as some manipulation of the immune response. So let's talk really quickly, briefly, the purpose of the immune system. It's primarily to fight infection, to prevent infection, and then also to prevent cancer. Today I'm really going to hit on the infection, but it's important to note that the immune system is also really essential for fighting cancer within our body. So let's begin with the innate immune system. The innate immune system can be described as energy efficient, rapid, broad-acting, nonspecific, and importantly, it is genome encoded, which means that every part of the innate immune system is coded by our genome and doesn't change over our lifetime or with exposure to a pathogen. This is important because it's in drastic contrast to the adaptive immune response. We'll talk about that later. So the innate immune response takes place between 0 and 4 hours post-exposure to some sort of a pathogen, and it's comprised of several components, primarily physical barriers, complement, specifically the alternative pathway, macrophages, and proteins. So there's some proteins that are generally secreted within mucus and the GI system, like defensins and pentaxins. The details aren't super important, but it's important to note that we have proteins that generally are toxic to pathogens, and that's part of the innate response. So we're going to begin by talking about complement because it's really essential to every part of the immune response. And so we're going to talk about some key facts of the complement. All complement are secreted by the liver as zymogens, and they're inactive. And complement proteins are going to be found in the blood, the extracellular fluid, and the lymph. So they're kind of ubiquitous in the body. There are multiple pathways by which complement gets activated, the alternative, mannose-binding lectin, and classical. But the one that's really important for the innate response is the alternative pathway. So we're going to quickly review the alternative pathway. The alternative pathway is composed of primarily 3 proteins, complement 3, factor B, and factor D. And the goal of many of these complement pathways is to activate what is called a C3 convertase, or essentially an enzyme that activates C3. So to quickly review, the alternative pathway begins with inactive or zymogen C3 in the lymph and soluble, and it's going to be reacting with water to form IC3. The importance is just that it's randomly happening within the solution within our body. So this IC3 just kind of exists. From there, factor B and factor D can act on that IC3 to form this, let me see here, sorry, I'm going to get a pointer. To get this molecule here, which is a soluble C3 convertase, meaning that this molecule can activate C3. So C3 can be activated to C3b and C3a. C3b then attaches to the pathogen surface. The important part of C3b is it then can be used furthermore to create its own C3 convertase. So factor B and D can bind to surface-bound C3b and form a second C3 convertase. In this way, by depositing a single C3b on a pathogen surface, you can coat the pathogen with C3b because there's this positive reinforcement pathway where one C3b can then become another C3 convertase. So anytime you have C3b on a cell, it itself can become a convertase and exponentially increase the amount of C3b. We'll talk about that with the other complement pathways, but just remember, C3b on a pathway, we'll use the alternative complement. But that's not where complement ends. The C3b, or the C3 convertase, which is this C3bbb protein, can actually attach to a second C3b molecule, and when this happens, it becomes a C5 convertase, and C5 convertase activates C5. C5 gets activated by the C5 convertase, and it gets cleaved into C5a and C5b. The important part is C5b will then move forward and interact with C6, C7, C8, and C9 to form what is called the membrane attack complex. This essentially punches holes into the membranes of pathogens and lyses them directly. All right, we talked about the pathway. Let's talk about the outcomes of complement. So complement by the convertases form C5a and C3a. These are called anaphylatoxins. In other words, they activate inflammation in the body and they recruit neutrophils. C3b, which is the protein that's bound to the cell surface, coats and opsonizes the pathogen, so the pathogen can no longer enter the body or enter a cell. So it prevents infection to start with. Additionally, there are receptors which bind to C3b, and this is a C1 receptor, C3 receptors, and CR4 receptors. These are important because any pathogen coated in complement is phagocytosed by macrophages and dendritic cells. So that complement really facilitates the phagocytosis process. And then finally, as we mentioned, the MAC complex directly causes pathogen cell death. So we should be asking ourselves, if complement is just existing in the body, in our solution, in our lymph, why don't our cells get attacked by complement all the time? And additionally, if complement is coating pathogens, how is it fast enough to do this? Well, that's because complement doesn't act alone. There's other factors that exist that help mediate complement. Those factors are factor H, factor I, propyridin, DAF, and MCP. We're going to go through each one of these. So propyridin is how complement is sped up. Propyridin stabilizes complement. So if propyridin is present, the C3 convertase is stabilized so that C3 can be activated in a much higher rate. This way pathogens can be coated in C3 complement much faster. Discussing the negative regulation or the prevention of self cells from getting coated in complement uses factor H and factor I. These are inactivating factors. So they come and work together to cleave C3b into IC3b. I think of this as inactivated C3b because if there is IC3b, it cannot become a convertase. So that stops this exponential pathway of coating. But that's not where our self cells end. They have another mechanism of preventing complement attack, and that's through DAF and MCP. So all of our self cells express DAF and MCP, and essentially what this does is it breaks apart the convertases. So if there's a convertase that forms on a self cell, DAF and MCP work together to break apart that convertase so the cell does not get coated. So it's kind of the regulatory mechanisms of the alternative pathway and complement. So this is a summary of just those pathways for you to look at later if you'd like. All right, so that covers the innate response. Now we're going to talk about the induced inflammatory response. This takes place from 4 hours to 4 days post-exposure. And it's primarily composed of macrophages with receptors that induce phagocytosis and inflammation, neutrophils, which are phagocytes, anaphylatoxins and complement, and we'll talk about those in a little bit, and then mast and basophils that degranulate and release histamine and leukotrienes. They just initiate inflammation. So the key part of the induced inflammatory response is it causes inflammation. So we're going to walk through the different components. But before we do, we're going to talk a little bit about the pathogens themselves, because pathogens have proteins on their surface that are obviously coating the pathogen, but they also contain proteins on the inside, the proteins that every pathogen makes. And so our immune response has evolved to detect both of those, and there are receptors that detect both extracellular and intracellular proteins. So we're going to talk a little bit about those. So the cell surface receptors on macrophages, there are many of them. The details, the names, aren't super important. What's important is to know that these receptors that are on the surface mediate phagocytosis. So if those receptors bind to some sort of a pathogen motif, the pathogen will be phagocytosed and degraded. I'm going to pause here and talk a little bit about pathogen motifs because that's kind of an ambiguous concept. So the immune system has evolved to be really efficient. When you have a pathogen that you're exposed to, you want to get a really fast response. So a way to think about this is to think, okay, if the immune system has to very rapidly figure out what is a pathogen and what is not a pathogen, it's going to evolve ways of just rapidly selecting for pathogens. And it does this by essentially having receptors to things that the body doesn't make. So if your body makes a protein, then it's a potential for it to be a self-protein. However, if your body, for example, does not make lipopolysaccharide, then anything that expresses lipopolysaccharide is therefore foreign. So the innate immune response motifs are usually directed at things that our body doesn't make, so that if they are found anywhere within us, they are by de facto foreign. So that's kind of what triggers this innate response. So let's go back and talk about a couple more surface receptors. So one surface receptor that is important that you should know and memorize is TLR4. This detects lipopolysaccharide, which is a cell surface receptor, or which is on the cell surface of a pathogen. And lipopolysaccharide binds to CD14, TLR4 dimers, and MD2. That complex is important to know. It will show up on exams later. But now let's talk a little bit about the intracellular receptors, because those also exist. So intracellularly, we have nod receptors. The names and the specifics are not that important. What's important is to know that the receptors detect proteins that are inside the pathogen. So that if a macrophage takes in a bacteria and the bacteria is degraded, its intracellular proteins are then going to be detected potentially by the nod receptors. And then nod receptors can induce inflammation. Both the TLR receptor and the nod receptor essentially work together through their individual signaling cascades to activate NF-kappa B. NF-kappa B is the primary inflammatory transcription factor that you should know. The pathway by which both of these receptors get to that point, not so important. The fact that they use NF-kappa B is important because it's going to induce inflammation. And so both the TLR4 and the nod receptor are going to induce NF-kappa B. So let's talk about, so what? So what do we get from NF-kappa B? Well, we get a lot of inflammatory proteins. This is a list of them. It's kind of like word vomit. So we're just going to cover a couple that I think are really important. IL-6 is a pyrogen. It induces fever, but it also induces acute phase reactant production by the liver. We're going to talk about this later, but acute phase reactants are really important. And then CXCL8 is a chemoattractant for neutrophils. So when macrophages release this, neutrophils are attracted to the site of inflammation and can partake in the inflammatory response. And then TNF-alpha induces vasodilation and ICAM production. Again, this is going to be important with neutrophils, and we'll talk about this in a few minutes. I've included some diagrams here on the slide, so if you would like to look at them later, you can kind of digest some of the details of these pathways. But the important part is that macrophages make inflammatory cytokines, and that induces inflammation, okay? So now let's talk a little about neutrophils. Neutrophils, aka polys by all the pathologists, are phagocytes, and they look like this. They're polymorphonuclear. And they also contain many surface receptors. Like with the macrophages, the names of the receptors, not super important. What is important is to know that if those receptors interact with the pathogen, it induces phagocytosis. Okay. Additionally, neutrophils, a key point of them is that they must migrate to the site of infection. So neutrophils are kind of existing within the lymph and the blood, and they're rolling around on endothelial cells, and they must somehow get a signal to move towards the infection. And that is where these signal proteins, CXCL8 I mentioned, and then the anaphytoxins, C5a and C3a, come into play. So when the anaphytoxins and CXCL8 are both released into the system, they kind of direct neutrophils. And the way they do this is they alter the receptors on the neutrophil surface so that as the neutrophil is rolling along the endothelium, it now acquires the ability to tightly bind and move through the endothelium via diapedesis. So the important part are that these inflammatory proteins allow neutrophils to migrate to the site of infection. If you don't have the inflammatory proteins, neutrophils can't get there and won't know what they're fighting. Neutrophils contain 3 granules. The names of the granules aren't that important. However, the enzymes they contain are somewhat important, and you should recognize them. So they have an enzyme called myeloperoxidase, lactoferrin, gelatinase, and then the essential one you must know, and I cannot stress it enough, you must know, is NADPH oxidase. What this does is it, bless you, it participates in what is called the respiratory burst. So the respiratory burst is essentially the neutrophil using oxygen to make toxic reactive oxygen species that kill pathogens. So if we look at the phagocytosis pathway, bacteria are phagocytosed, and they're exposed to these granules. When they're exposed to the granules cause reactive oxygen species, which damages the pathogen. And that is essentially the quintessential mechanism by which neutrophils kill pathogens, is with these very reactive species and very strong enzymes. Now, following the release of their granules, neutrophils actually commit apoptosis. And this is because neutrophils can't recreate granules. When they develop, they make the granules. Once they use them, the neutrophil must perform apoptosis. And this is important because if you imagine a neutrophil has taken up a pathogen and degraded it, if for whatever reason that pathogen has survived, by performing apoptosis, it's one more measure in hopes that that pathogen will be destroyed. And additionally, macrophages will then come and consume that whole conglomerate of cell and byproduct. So I want to really highlight NADPH oxidase because it's super important. And essentially, this part of this respiratory burst. So it's going to produce superoxide, which can then be used by superoxide dismutase to make hydrogen peroxide. These are the reactive oxygen species which bacteria are going to be exposed to and other pathogens. As you move forward, you will see a disease called chronic granulomatous disease that is the loss of this superoxide, or this NADPH oxidase. So really watch out for that and really think about it's a neutrophil problem. All right, we're going to return to complement. I said complement's involved in everything. So now we're going to talk about complement from the mannose-binding lectin pathway. But before we do, recall that in the induced inflammatory response with macrophages, acute phase reactants are created by the liver. This is mediated in part by IL-6. It turns out one of these acute phase reactants is actually mannose-binding lectin. And so mannose-binding lectin can then bind to cell surface and activate complement. So it's kind of this feed-forward mechanism. It's a common theme of activating the immune response and feeding forward. So through it. All right, so mannose-binding lectin looks a lot like this. It looks kind of like an upside-down bouquet of flowers if you squint your eyes and tilt your head. And what it's going to do is it's going to bind to the cell surface of a pathogen binding to mannose. When it does this, it has serine proteases, these MASP1 and MASP2. Name's not important. The important part is that this mannose-binding lectin can activate C4, which is another one of the complement proteins within our lymph and our blood. When C4 is activated, it gets deposited on the membrane surface. The mannose-binding lectin can then also activate C2 and attach C2 to C4. Note, the nomenclature for C2 has changed, and it's now C2b that attaches, so the textbook image is just incorrect in that it's A instead of B, so just nomenclature. The important part is that this molecule here, C2b attached to a C4b, is a C3 convertase. So this is now the third C3 convertase we've learned. And what does a C3 convertase do? Well, it deposits C3b on the surface. Now, what did we talk about when we talked about the alternative pathway? Anytime you have C3b on the surface, the alternative pathway can be used to coat the pathogen in C3. So we start with mannose-binding lectin. That makes a C3 convertase, which deposits C3. Then that C3 goes back to the alternative pathway and can work with factor B and factor D to coat the pathogen in C3b. So you start with this mannose-binding lectin, and that leads to the alternative pathway. It's really how this system exponentially creates inflammation and coats these pathogens and opsonizes them. But that's not all. C-reactive protein also interacts with complement. C-reactive protein is also an acute phase reactant. So the C-reactive protein can attach to a different complement receptor or complement protein, C1. C1, just like mannose-binding lectin, activates C4, and then C4, and then it also activates C2, which makes that C2-C4 C3 convertase. So the mannose-binding lectin pathway and the C-reactive protein pathway are almost identical. It's just a matter of whether it's C1 binding or mannose-binding lectin. Either way, they lead to deposition of that C3b and then the propagation of that C3. All right, so we've talked a lot about complement. What about viruses? Because viruses are kind of a unique thing. They're not really alive. They're not really dead. They're just packets of DNA. So viruses infect healthy cells. And these healthy cells, all they do is express virus proteins. So the immune system needs to be able to monitor whether or not viruses exist. So we have several mechanisms by which this is done. The first one is the interferon response pathway. If a cell detects virus protein within it using RIG receptors, it'll activate interferon production. Interferons essentially just tell the cell and all cells surrounding it, hey, there's a virus, watch out. And they essentially are going to make the environment of the cell inhospitable. So they're going to reduce protein production, they're going to reduce DNA reproduction, because the goal is to reduce viral spread. So that's the interferon response. There's also natural killer cells. And natural killer cells are part of the induced inflammatory response in that they can detect the loss of an MHC on a cell surface. We'll talk about MHC later, but MHC should be present on every cell. And if it's missing, it's a sign that something is wrong. And if something's wrong, NK cells will then recognize it and attack. They can induce target cell apoptosis, and they also induce inflammation in the surrounding tissue. They have TLR receptors, but these receptors are specific for DNA and RNA, which are the products of viruses. And then finally, they are carefully regulated. Because they're cytotoxic cells, you don't want them just running around willy-nilly and killing your good cells. So they're highly regulated. All right, so let's take a quick look at this. Again, a virus can infect a cell, and it's going to produce protein. It's going to produce RNA, DNA, and proteins virally. And what we're going to have is activation of Rig receptors that produce the downstream effect of interferon alphas. Interferon alphas essentially act paracrine and autocrine. This induces the interferon alpha response. So nearby cells are signaled by interferons that, hey, there's a virus, and they become inhospitable. Let's talk briefly about natural killer cells. The natural killer cells essentially run around in our lymph nodes and in our blood, and what's going to happen is if they are signaled that there is a virus present, they will then move to those virally infected cells and induce them to perform apoptosis. So they are cytotoxic cells that broadly notice when something's wrong and induce that cell to perform apoptosis. Natural killer cells aren't going to be highly tested on. Just know the concept that they're a cytotoxic cell that's patrolling the body. All right, so now we've exhausted the induced inflammatory response. Let's talk a little bit about the adaptive immune system. So the adaptive immune system is highly specific. It's only partially encoded by the genome. We'll talk about the importance of that later, but it's driven by random mutations, and random mutations are essential. It's a slow response, so it takes a little more time than the innate response, and it's also going to have both extracellular and intracellular pathways because pathogens can be both extracellular and intracellular. So let's talk about the essential components. The adaptive immune response is composed of macrophages, dendritic cells, MHC molecules, B cells, antibodies, and T cells. We're going to walk through each one of these. But we're going to begin with B cells. B cells produce antibodies. That's their purpose in life. They just produce antibodies. And this is part of the humoral response. A little bit about antibodies. They bind to proteins, polysaccharides, and even inorganic matter. So they're really broadly acting. They're present in the blood, lymph, mucus, and other bodily fluids. So just like complement, they're kind of ubiquitous everywhere. But a key point about the B cells is that the B cell receptor is a surface-bound antibody. And every B cell makes a single B cell receptor. So we have a lot of antibodies because we have a lot of B cells. We don't have multiple B cell receptors on one B cell. Right? So every B cell makes one type of antibody, and that's it. The variability comes from having multiple B cells. And then additionally, an activated B cell that makes antibodies is called a plasma cell, just for terminology. All right, some basics about antibodies. Antibodies are kind of described in multiple ways. So some components of them are light and heavy chains, where you have a heavy chain that is longer and a light chain that is smaller. Variable and constant regions. We're going to talk about the importance of these in a little bit, but know that they exist, and it's a way of classifying the locales of an antibody. And then FAB and FC fragments, the fragment that binds to an antigen and the constant region. All right, so in addition to having different ways of describing antibodies, there's different classes of antibodies. There's 5 of them, G, M, D, A, and E. And the class is determined by the constant region, okay? And so by changing the constant region of the antibody, you change the class of the antibody. Anywhere that an antibody binds is considered an epitope. So on the side here, we've got 4 different epitopes with 4 antibodies binding. It's for some terminology. And then we can now elaborate a little bit more on the variable region. So if we look at the FAB segment, the very tip of that segment is where the antibody will bind to its epitope. And the reason this is important that it's highly variable is because pathogens are really variable. Pathogens can have just about any epitope you can imagine. So you need to be able to make an immune factor, in this case an antibody, that responds to all of these different options. So just like the antigens here are different shapes, the ends of an antibody are highly variable, so they can attach to very different epitopes. And then you have a constant region because once you've tagged something, the immune system needs to be able to recognize it. So if an antibody tags a pathogen, all the immune cell needs to know is that a pathogen is tagged. So by having a constant region, no matter what the antibody attaches to, immune cells can detect that constant region and recognize it as an antibody. All right, let's talk a little bit about antibody development and how antibodies are made. Antibodies essentially address a problem in the body. And our problem is that there are thousands of pathogens, more than thousands of pathogens. But we have a finite genome, which means we can't have a separate gene for every pathogen. It's just inconceivable to have that many genes. There's a second problem. Most proteins that a pathogen makes, we also make, which means that the immune system needs to be able to recognize foreign from self and needs to be really highly specific. So, as with many things, evolution has a solution for a lot of these problems. We have evolved multiple gene segments. The way I like to think about this is kind of like choosing your outfit in the morning. An outfit has a shirt, pants, and socks. Really plain outfit, but an outfit nonetheless. And to make a functional outfit, you need one of each of these. And antibodies have gene segments, V, D, and J, where the D region is only present on a heavy chain. So if we look at this in pictorial form, we can see that the V regions are red, the J regions are yellow, and the D regions are green. And if we use our analogy, we can say that the red region is a pant, the green region is a shirt, and the yellow region is a sock. To make a functional antibody, you need one of each of these, okay? So if we look at this, what happens is you essentially take one of these, you choose one of these, choose one of the shirts, choose one of the socks, put it together, and you get a functional variable region of your antibody. We can look at a larger description of this and see that when you've chosen one variable, or one V, one J, or one V, one D, and one J, you get this region on the variable part of the antibody, so right here at the tip where it's going to bind to a pathogen, that is composed of one of each of those possible combinations. So this process of choosing one of these different segments is called somatic recombination. Okay? So it's essentially the antibody choosing its outfit. So how is this done? Well, it's done with a protein called REG. REG comes in and essentially just cleaves the gene between two of these little segments. So it takes a V segment and then takes a J segment, cleaves everything out in between, and lines them up next to one another so that you can get, when the polymerase moves through, it hits a V region, then a J region, and nothing in between. All right? But that's not all. There's always a little bit more. And so when this is done, if we look at some of the process of it, you get additional diversity by a protein called TDT. So if you imagine you've got your D fragment you're going to use and your J fragment you're going to use. The REG protein is going to come in and it's going to clip all the genome in between. So that's the orange, is everything in between. When it clips it, you get two blunt ends, your D segment and your J segment, and you want to bring those together. So you're going to open it up and get ready for the DNA repair enzymes to bring those together. But at this point, random nucleotides are inserted at the ends. This random nucleotide adds variation to this region. So remember, once this is put together, it's going to be transcribed as D through J. And so all of this region in between is random. There is no gene that encoded that. So that's where the adaptive immune response is sort of encoded by the genome, because the D segment and the J segment came from the genome, but this bit in between was randomly generated. So that's really important for allowing antibodies to bind to a very broad list of epitopes. So after the random mutations are inserted, essentially that gene sequence in between is filled in, and you now can make a functional D and J together. All right. So now we've talked about that a little bit. Let's talk about kind of what that means and the concept of that. So if we look at the amount of combinations we can make based on the number of V, D, and J segments, just in the heavy chain, you can make 5,000 combinations. And just in the light chain, you can make 200 combinations. And of course, an antibody has heavy and light chain. So if you rearrange those a little bit, you get a total of about 1.6 million different combinations of antibodies you can make in total. Notice that you don't have 1.6 genes in your genome. Instead, you have these little fragments that can be shuffled together from somatic recombination. So this is a neat trick that evolution has devised in order to give you a lot of diversity, a lot of bang for your buck. All right? So let's talk a little bit about antibodies. Antibodies come in different classes. And these classes are going to be determined by the placement of the constant region in the genome. All right? So all naive B cells express IgM and IgD. We can explain that by looking at the genome. if you imagine that polymerase is going to come through, it's going to transcribe everything as it goes along, right? And it turns out the first thing it's going to hit is this C mu, or the constant region for the M type of an antibody, okay? So the reason that a B cell that's naive makes IgM and IgD is because those are located first on the genome. And so that's the first thing that's going to be produced, all right? The polymerase won't ever make it out to here because it hits a constant region first, all right? So that's why we have IgM and IgD expressed on naive B cells. If we look at a B cell receptor, as I mentioned earlier, is a surface-bound antibody. And it's essentially just combined with Ig beta and Ig alpha because these stabilize that surface-bound antibody. Without these, that antibody wouldn't be stable on the cell surface. All right, now we've talked exhaustively about antibodies. What about B cells? Let's talk a little bit about B cell development because B cells must come from somewhere. So B cell development happens in the bone marrow, B and B. Doesn't happen anywhere else. And negative selection of B cells also happens in the bone marrow. All right, the next couple slides are going to be busy in appearance. That's just because the images are confusing looking. We're going to go over big picture and the importance of them so you don't get bogged down in the details, okay? So the first step in B cell development is going to be the formation of a heavy chain. So we talked about how all those segments are recombined. When they're recombined, the protein will be produced. If that heavy segment that's produced is functional, great. That cell will proceed to step 2. If when those segments are recombined and that TDT inserts random mutations, if that new gene is not functional and doesn't fold properly, it can't proceed to the next step because it doesn't make a functional heavy chain. So instead what it does is it rearranges the second chromosome because we have 2 chromosomes conveniently. And if that fails, the cell will stop developing right then and there. The B cell has to have a functional heavy chain before it can proceed. All right? So step 2 is that the B cell must produce a light chain. And a light chain goes through the same process, recombination, production into a protein. If that protein is functional, great, the B cell moves on, right? So the concepts to remember is in development, you make a heavy chain, then you make a light chain. If both are properly folded and functional, you go on to selection, okay? So next we're going to talk about negative selection because that's the final step in the maturation process. Negative selection for B cells happens in the bone marrow. And let's talk a little bit about what negative selection is. Negative selection is part of what we consider to be central tolerance. Your immune system is designed to fight pathogens, which means you don't want your immune system fighting your own body. So there needs to be a way to control this because with so much randomness happening in the generation of a receptor, the odds are you're going to get receptors that act against your proteins that you make. So the body has to have a way to control these rogue B cells. The way it does this is through negative selection. So in the bone marrow, after a successful B cell receptor is made, the B cell will be exposed to epitopes that your body makes, your self-epitopes. If that B cell reacts to any of those, it is not allowed to go into circulation. Instead, what happens is the light chain is rearranged yet again, okay? So the B cell will continue to make a new B cell receptor until either that receptor does not react with the self antigens, or the B cell no longer has any genes it can recombine because it's already spliced out too much of its genome, okay? If the B cell is unsuccessful and continues to react to self-peptide or self-epitopes, the B cell will perform apoptosis and won't be released in circulation. If the B cell successfully makes a receptor and does not react to anything in the bone marrow, it passes negative selection and moves into the lymph. Once in the lymph, the naive B cell will move through the lymph nodes through the rest of its life looking for something that reacts to its receptor. So that is the pathway of B cell development. Starts in the bone marrow, goes through negative selection, gets released into the lymph, but then patrols all of the lymph nodes looking for an epitope or an antigen to bind to. All right, so that takes care of B cell development. What about T cells? Well, T cells are part of the cell-mediated immunity. They require MHC, and there are 2 types, CD8, which are cytotoxic, and CD4, which are helper cells. Before we can talk about T cells, though, we must address MHC, and because MHC is super important when it comes to T cells. So MHC binds proteins. It's key. It only binds proteins, and it's highly specific. All right, But why do we need MHC? Because I think it's important that we try and understand what the function of MHC is. Well, if you imagine yourself as a T cell, your goal in life is to patrol the body looking for something that is a pathogen. The question is, how do you find that pathogen? And the answer to that is MHC. MHC being expressed on every cell is essentially a hand holding up an antigen for T cells to come and patrol. So all the T cell needs to do is interact with the different cells in the body and look and see if there's something it recognizes on the MHCs. So that's kind of the purpose for MHC. It's a way of displaying antigens for T cells to patrol. All right, there are two classes of MHCs because you have extracellular proteins and intracellular proteins. So MHC class 1 is for intracellular proteins, and these are fragments that are derived from the proteasome. The MHC1 is expressed on all nucleated cells except for syncytial trophoblasts. This is just a fun fact because syncytial trophoblasts are involved in pregnancy. You don't want an immune response against the fetus, so you don't have MHC1 on syncytial trophoblasts. And MHC1 class 1 interacts with CD8 T cells. The other class is MHC class 2, and this is for the extracellular protein pathway. And the fragments for this are derived from lysosomes instead of the proteasome. And MHC class 2 is present only on antigen-presenting cells and interacts with CD4. So let's quickly review these pathways. MHC class 1 begins in every cell in the body with the proteasome. Peptides are broken down by the proteasome into fragments about 8 to 10 amino acids in length. From there, they're transported to the endoplasmic reticulum using TAP. Once in the endoplasmic reticulum, they can bind to MHC class 1. When MHC class 1 has a peptide fragment bound, it moves to the Golgi and then is exported to the surface where CD8 T cells can come and monitor those peptides. Okay? That's MHC class 1. I've included a pictorial diagram on here for you to look at later if you so choose, but now we're going to move to MHC class 2. MHC class 2, as I mentioned, is part of that extracellular pathway. So you have peptides here in blue that are in the extracellular space, and you have antigen-presenting cells. Antigen-presenting cells go through, and they're going to endocytose material from the environment. That material will be exposed to the lysosome, and in the lysosome, that material will be broken down. If that material can bind to an MHC class 2, the MHC class 2 will then be exported to the cell surface, where now will be monitored by CD4 T cells. Okay? One point that I want to bring up is that there's something called the invariant chain, which sits in the MHC class 2 during all of its development and prevents rogue self-proteins from binding in the endoplasmic reticulum. So the invariant chain is removed in the lysosome so that lysosomal products can then be added to the MHC class 2. That's just a way of making sure that no self-proteins are put on it. Okay? Here we can look at this visually. This is the invariant chain. Again, I mentioned in the endoplasmic reticulum, it's added so that none of these proteins in the ER can get onto MHC class 2. Once in the lysosome, the invariant chain can be removed because now everything it gets exposed to came from the extracellular environment. All right? I would be remiss if I didn't address the population variation in MHC, because MHC, each of us has a set number of alleles of MHC. So if our MHC doesn't respond to a certain peptide, we can't make an immune response against it, at least a T cell immune response. But the population has a lot of variability. These MHC alleles, you notice their variability is right there where the peptide binds. So even though we are restricted in that we only have a couple of alleles of MHC, throughout the population, there's a lot of variation in the MHC that is presenting peptide fragments. For all of you, there's a slide that summarizes where MHC is and compares MHC class 1 and class 2. For time purposes, I'm going to keep moving. All right, let's now talk about T cell receptors. Because we've talked about MHC, now we can talk about the T cell receptor. It's very similar to the FAB segment of an antibody. In fact, if we look at it, here's an antibody, here's the FAB segment, here's a T cell receptor. It looks almost identical, except for instead of having a heavy and a light chain, it has an alpha and a beta chain. Otherwise, its development, its production is very similar. But we'll go through it just for posterity's sake. So the T cell receptor is going to bind to MHC that contain peptide fragments. So T cell receptor is binding to an MHC with that red peptide fragment. Bless you. And importantly, the T cell receptor requires the co-receptor CD3. CD3, just like with the B cell receptor, helps stabilize this surface bound receptor. So you need CD3 to have a stable T cell receptor. And then T cells have CD4 and CD8, which are the co-receptors, and determine which class of a T cell is going to be made. So CD8 interacts with MHC class 1 because it interacts with this alpha 3 domain, while CD4 interacts with MHC class 2 because of this beta 2 domain. It's not important to know all the domains. What's important to know is that the co-receptor determines which class of MHC that T cell can interact with. All right, let's talk about developing this receptor. The receptors also have these V, D, and J segments, just like the antibodies did. And the process for developing them is very much the same. You make an outfit, and that outfit becomes a T cell receptor. So you're going to choose, the body chooses a V region, a J region, and if you're a heavy chain, you also get a D region to choose from. And then it's going to combine those using the protein RAG into a functional variable region, all right? And so that is how T cells are formed. It's analogous to how B cell receptors are formed, okay? So let's talk about a term called MHC restriction because it comes up a lot and it's really important. T cell receptors require a specific binding to an MHC and a specific binding to a peptide fragment. So if you have a T cell receptor and you change the MHC, that receptor will not bind to it because the receptor is specific for a given MHC. Likewise, if you have the same MHC, so in this case the yellow MHC, but the peptide fragment changes, the T cell receptor will not bind. So it's selective for both the MHC and the peptide within that MHC. And that combination of selectivity is called MHC restriction. All right, now we've talked about the T cell receptor, let's talk about the development of T cells. So development of T cells happens in the thymus, T cell thymus, okay? And positive selection is going to happen in the cortex. Negative selection is going to happen in the medulla. And we're going to walk through this process just like we did with B cells. So the first step, again, analogous to B cells, is the rearrangement of the beta chain. Beta chain is just like the heavy chain in the antibody. So you're going to get, there has 2 attempts to make a functional beta chain. If the beta chain is made, the T cell will proceed to step 2, where step 2 is alpha chain production. Here, the T cell will attempt to recombine its genes to make a functional alpha protein. If a functional alpha protein is made, the T cell will continue on development. If at either step, either the beta or the alpha chain fails to make a functional protein, the T cell will attempt to recombine those genes. If it can't, it will perform apoptosis. So T cells are controlled so that only those with functional T cell receptors move on to selection. All right? So then, the third step. The third step are called mature double positive cells. So at this point, you have a T cell, it has a functional receptor, and that T cell is going to express both CD4 and CD8. And this is called a double positive T cell. This is essential because the next thing that's going to happen is the T cell is going to go through positive selection. This happens in the cortex of the thymus. Positive selection is where T cells interact with MHC. As I mentioned, T cell receptors are MHC, they're MHC selective, right? So a T cell must recognize MHC because that's what T cells are looking for. So in positive selection, the T cell receptor is exposed to MHC class 1 and MHC class 2. And the whole goal of positive selection is to make sure that that T cell recognizes an MHC. If the T cell doesn't recognize the MHC, it's not a functional receptor and that T cell undergoes apoptosis, okay? If that T cell recognizes an MHC, that's a good thing because that T cell knows what it's looking for and will proceed through the developmental pathway, all right? So positive selection is selecting for MHC, okay? At this point, the T cell will become single positive. The way this happens is that when a T cell receptor interacts with an MHC, it doesn't matter during positive selection whether that MHC is class 1 or class 2. It's just a T cell receptor interacting with MHC. If that T cell, for example, interacts with MHC class 2, then the CD4 co-receptor will be preserved because that is required for that T cell to maintain the activity, and CD8 is no longer needed, okay? So the type of T cell, the class of T cell, is determined by positive selection and which MHC the T cell interacts with, all right? So now, we have a T cell that has a functional receptor. It knows that it's looking for MHC, and it moves on to the process called negative selection. This is, again, a component of the central tolerance. central tolerance being that we cannot have T cells that react to our self-proteins. That would be horrible. So we have negative selection, where this amazing protein, and I think this is like the coolest part of immunology, kind of a nerd when it comes to this, but there's a protein called AIRE, A-I-R-E, that's made by dendritic cells in the medulla of the thymus. A-I-R-E, or AIRE, its only job is to just transcribe a bunch of the proteins that the body makes. So the goal is that when T cells interact with these dendritic cells in the medulla, they interact with almost every self-protein that we have in our body. So AIRE is like a universal transcription factor that's designed to expose T cells to all of our self-proteins that we have, okay? In negative selection, T cell binds to any of these fragments. That means that the T cell is binding to a self-peptide, because self-peptides are expressed by these dendritic cells. If that happens, that T cell cannot go into the periphery because it will be self-reactive and performs apoptosis, all right? If that T cell goes through the medulla of the thymus and doesn't react to anything, that T cell will proceed to be released into circulation and will move through the lymph nodes just like B cells do, patrolling for its antigen. All right? So negative selection, both with B cells and T cells, is a control measure so that we don't have any self-reactive cells. Or at least that's the hope. That's the goal. All right? So now we've talked about B cell development and T cell development. We've talked about antibodies and T cell receptors. Let's finally get to the activation of the B cells and the T cells. All right. We're going to start with the CD4 T cells, but a lot of the concepts of activation apply to all of the adaptive immune response, okay? Activation of the adaptive immune response, be that a T cell or a B cell, requires two signals. It requires its T cell receptor for a T cell and some sort of a co-activator signal, okay? For T cells, this co-activator is B7 and comes from dendritic cells or macrophages. So we're going to look at this quickly with a little animation. So T cells, okay, here we go. So a macrophage that is activated will produce MHC class II on its surface. If a T cell comes and interacts with that MHC class II, the first signal is activated. It needs that second activation signal, and that comes from B7. An activated dendritic cell or macrophage expresses B7 and gives the T cell its co-activation signal, or its second switch. So whenever you're thinking about activation of a T cell, think that it needs two switches, or two signals. One is the T cell receptor pairing with an MHC, and the second is a co-activation signal. In this case, it's B7. Right? You always need those two things to be active. All right. We can look at this pictorially, that you have signal 1 is the pairing of a T cell receptor with an MHC combined with a peptide fragment. So that's your signal 1. And all of these signals work together in a downstream signal cascade to induce transcription factor activity. So I apologize for the very confusing slide. The important part is the downstream activators are NF-kappa B, NFAT, and AT1. They're transcription factors. If the T cell is activated, transcription factors are activated that cause that T cell to reproduce and to become, start expressing all of its activated enzymes, okay? So you don't have to know the entire pathway, but it's important that you know that activating the T cell activates these very key transcription factors, all right? So here this is just showing that you need this B7 co-stimulation to get T cell proliferation. Of note, this receptor here on the T cell surface has some variations. So CD28 is an activation receptor. If CD28 interacts with B7, it activates the T cell. But there's a couple variations on the theme. If CTLA4 interacts, it's a deactivation signal. And then if PD1 interacts, it induces apoptosis. So these are just variants on the theme where the T cell receptor could still be interacting, but the co-receptor will determine the fate of that T cell. All right, there's one more thing that T cells need to be active, and that's IL-2. IL-2 is kind of the growth factor, the inflammatory factor that T cells require to be fully active. So a T cell that's activated will produce IL-2. IL-2 will go into circulation and will act both paracrine and autocrine to activate the T cells that are exposed to IL-2. it's essential. If IL-2 is missing, you don't get T cell proliferation near as strongly, okay? So IL-2 is this like T cell activation factor that is produced by T cells to induce their own reproduction and their own proliferation. All right, so let's talk quickly about subtypes of CD4 T cells. There are 5 subtypes. It is important to know them, but I'm going to focus on 2 just for the sake of time. The ones that I'm going to focus on are Th1 cells and T follicular helper cells. So we're going to talk about the effector function of these CD4 T cells. So the first function that's important with CD4 T cells is activation of macrophages. So a T cell that's activated will produce something called CD40 ligand. So a T cell gets activated, it makes CD40 ligand. CD40 ligand interacts with macrophages and activates macrophages. Now this is really interesting because it's taken the adaptive response and fed back all the way to the innate response. So by activating these T cells, you go all the way back to activating macrophages and helping to proliferate inflammation and phagocytosis. All right, the second function of CD4 cells that we're going to talk about today is activation of B cells. This also uses CD40 ligand. So an activated T cell makes CD40 ligand. If that T cell interacts with a B cell that has the MHC that corresponds to that T cell receptor, and the T cell is active, it will provide the B cell with CD40 ligand. CD40 ligand fully activates B cells. We'll talk about this when we talk about B cell activation. But CD40 ligand is essential for activating B cells to their full potential. All right, so now we're going to briefly talk about CD8 T cells because they have slight variations on the theme. While CD4 T cells require just 2 signals, CD8 T cells can sometimes require 3. Sometimes they can require a CD4 T cell. The reason for this is that CD4 T cells are helper cells. They just kind of go around and help activate other cells and help produce inflammation or regulate the immune system. But CD8 T cells are cytotoxic, which means that like once a CD8 T cell is activated, it's the end-all be-all and it will go on and kill cells. So you want to make sure that you have tight control over the cytotoxicity. So sometimes the activation of CD8 T cells requires the T cell receptor, the co-activator of B7, and a CD4 T cell. So that's just a small variation on the theme for the B cells. And then, as I mentioned, B cells are cytotoxic. So once a B cell is activated, if it interacts with any cell expressing an MHC that is conjugate to that T cell receptor, it will induce that paired cell to perform apoptosis. So the idea is that T cells get activated by a specific MHC, and then any time after that, if they interact with that MHC peptide combination, they will kill that target cell, assuming that it is foreign. So this is why we don't want T cells to become rogue, or CD8 T cells to become rogue, because they will start recognizing that MHC peptide complex. All right, time to talk about something called peripheral tolerance. We talked about central tolerance with negative selection. Now we're going to talk about peripheral tolerance. So peripheral tolerance is where if you have a T cell that interacts with a T cell receptor but does not interact with its second co-activation signal, that T cell becomes anergic. In other words, it's assumed that if the T cell interacts with an MHC peptide but doesn't get that co-receptor, then that peptide was probably a self-peptide and you don't want that T cell to be active. So it becomes anergic. After that, if it's exposed to its co-receptor, it remains anergic because it's still assumed that it was self-reactive. So this is a way, again, of controlling for any self-reactive T cell. So if the T cell makes it out of the thymus but is still self-reactive, it can become anergic in the peripheral or in the lymph system. All right, that takes care of B cells. That takes care of T cells, my apologies. Now it's time to talk about B cells. B cell activation, same theme, requires 2 signals, the B cell receptor and a co-activator. In this case, the co-activator is CD40 ligand from T cells. We mentioned this when we talked about T follicular helper cells. So let's take a look at a quick animation of this. So if we have a B cell in the lymph node, the B cell can interact with the antigens that are present in the lymph node. If it interacts with an antigen and the B cell receptor reacts to it, it'll activate the first signal, the first switch. If it then subsequently interacts with a T cell and an MHC combination here, if it interacts there, it then gets its co-activation signal from CD40 ligand. Now remember, this CD4 T cell is an activated T cell, so that's why it produces that CD40 ligand. Okay? So a B cell requires activation from a B cell receptor and then co-activation from an activated T cell. Okay? So that is how a B cell becomes fully active. We can look at this through some diagrammatic images. That there are variations on the theme, but activation of the B cell receptor is your first switch. That happens out in the cortex of the lymph node. The B cell then interacts with T cells that are activated T cells. These are going to give that co-activation signal of CD40 ligand. This happens in the T cell region. And then finally, the pair of them, the B cell and T cell, will move back to the cortex where they will form what is called a germinal center. So we can take a quick look at this through an animation. If we have a dendritic cell in the periphery, the dendritic cell will pick up the pathogen and will become activated. When activated, the dendritic cell will express a protein CCR7. This will allow the dendritic cell to migrate to the HEV in the lymph node. This is the entry site for T cells and some B cells. Okay? Once inside the lymph node, circulating naive T cells will move around and scan the environment. So naive T cells express CCR7 on their cell surface, which attracts them to the lymph node. So now you've got a dendritic cell and T cells all in the lymph node. T cells will scan the dendritic cells that are in the T cell region of the lymph node until they meet their pair, until they meet a MHC that reacts with their T cell receptor. At this point, they will get their first signal and then their second signal, because remember, that's an activated dendritic cell. So now that T cell becomes activated, it gets its two signals, and it begins to proliferate. And as it does this, it also begins to move towards the cortex of the lymph node. All right? So an activated T cell in the lymph node proliferates and moves towards the cortex. So now we're going to talk about B cell activity, because this is happening simultaneously. So in the periphery, we have macrophages and dendritic cells. These macrophages and dendritic cells are going to use CR2 to pick up pathogens and take the pathogens to the lymph node. They're also going to take just any sort of antigen to the lymph node. These antigens are added to follicular dendritic cells, and then B cells migrate to the lymph node and patrol these follicular dendritic cells, looking at all the antigens on the surface. If the B cell reacts to one of these antigens, it gets its first signal, and it takes up that antigen, and then it can either become an IgM secreting plasma cell that's immature, or it can then proliferate and begin moving towards the T cell region. Remember, at this point, it's only had its first signal activated. It's only had its B cell receptor. The T cells and the B cells meet in the middle because they're migrating in opposite directions. And if the B cell expresses an MHC that with a peptide fragment that matches the activated T cell in that region, they will combine to form a conjugate pair. And you will see this CD40 ligand interact with the CD40 receptor on the B cell, giving the B cell its second activation signal. The pair of them move together towards the cortex, where they form a germinal center. All right? And in this germinal center, the B cell and the T cell will work together to mature and continue to make antibodies and perfect the antibody. All right? So let's talk about how this happens. In a germinal center, you have two processes that happen. You have class switching, and you have somatic hypermutation. Both are essential to make high affinity antibodies. All right? We're going to start by talking about class switching. Class switching, remember we talked about how the genome, you've got all these constant regions lined up in your genome. Class switching uses AID, and essentially all it does is the AID protein comes in, cleaves out part of the genome in a constant region, removes it, and combines the genes so that now instead of having the IgM and IgD, you can make IgG. So essentially it just removes the genes that are in between and brings a constant region closer. We can look at this on a larger scale here. So you've got your VDJ of your variable region of that antibody. Then you can see your constant regions lined up. AID just removes the region of genome between a constant region and the variable region so that you have a new constant region that's transcribed. All right? Essentially, you're just shortening your genome so you get a new constant region. All right. It's important to note that because the genes are actually removed, you can't go backwards. So once you've class switched, you can't re-add that gene fragment back into the genome. So B cells cannot reverse class switch. They can only continue to class switch by cleaving out further parts of the genome, all right? So I've included here a list of all the classes, kind of where they're important and what they're important for. We're not going to go over it now just because of time, but you might want to take a look at it and just familiarize yourself with the different classes of antibodies. We are going to touch on two of them. First one is IgM. It's important to note that it's a pentamer. We're going to talk about it a little later. And the second one is IgA, and this is secretory. So IgA is important for secretion of antibodies into the GI system, for example. All right, now let's talk about somatic hypermutation. Somatic hypermutation is mediated by the AID protein, just like class switching was. And this is also a really cool process because what AID does is it goes in and it makes random cuts in the DNA so that it induces random mutations in that B cell receptor. You may wonder, why would you want to do this? Well, if you imagine you've got a B cell receptor that you know reacts to your antigen because the B cell is active. But remember when we talked about the problem being that our immune system needs to be highly specific? So if you know that that B cell reacts with an antigen and you want to make it better, you want to do this incrementally. You don't want to just change everything all at once. So if you induce a couple random mutations, there's a probability that that mutation will cause stronger binding. If that happens, that's great because you made a higher affinity B cell receptor and a higher affinity antibody. However, that hypermutation can also go the other direction, where random mutations can cause the receptor to be less strongly binding. In the process of somatic hypermutation, if the mutations cause that receptor to bind more tightly, the receptor will continue to be made, and that B cell will move forward and become a plasma cell. If that receptor binds with less affinity, that B cell will not move forward and will undergo apoptosis. So somatic hypermutation in the germinal center is the way by which B cells make really high affinity antibodies, all right? And again, that's from random mutations. So this is the second part in antibody production that's not genome encoded. It's random mutations. All right, and as I mentioned earlier, mature B cells are plasma cells. Once they have made high affinity receptors, they then internalize that and secrete it as an antibody, and that is considered a plasma cell. All right, we're almost there, I promise. The last little bit of material we need to cover is the classical pathway. And this is mediated by antibodies. So, this is the complement classical pathway. So as I mentioned, complement is involved in the entire immune response. And this is amazing that now we have a part of the complement system that responds to antibodies. So how does this happen? Well, that IgM that I mentioned, it was a pentamer, can bind to its epitope on a pathogen surface. When it does that, C1 can bind to the IgM. Remember all the way back from that C-reactive protein pathway? C1 can activate C4 and C2 to make a C3 convertase. Once that happens, you make C3b. When you get C3b, you activate the alternative pathway, and you coat that pathogen in C3b. So this classical pathway is kind of like the culmination of all your immune system's work, because now the antibodies are activating complement to then go back and coat these pathogens in complement proteins. Of note, this also can happen with IgG. So IgG can also attach to the surface of a pathogen, and when a bunch of them are attached, they can interact with C1, and they can use the same pathway to activate the complement cascade. All right, one final note. The classical pathway using IgG is the way that our body clears the antibody-antigen complexes. So imagine that you have a little peptide in your blood and lymph that an antibody has attached to. If you don't remove that from the blood, eventually you would clot and it would deposit in the membranes and it would be a disaster. So somehow you need to remove the complex of an antibody with an antigen. And the way that's done is through complement. So complement comes in using C1 and adds a C3b, just like the pathway we talked about. From there, red blood cells have CR1 receptors, take in that antigen complex, and they're going to give it to macrophages to eliminate from circulation. So not necessarily important that you memorize all the steps of this. What is important is to recognize that antigen and antibody complexes are cleared using red blood cells and macrophages. because there are pathologies that are a result of too much of this antigen-antibody complex, all right? So let's put it together from start to finish. We're just going to summarize what we talked about, okay? So from start to finish, we begin with the innate response at the periphery. The innate response happens when a pathogen is exposed in the periphery. The innate response is usually going to be your barrier. If that fails, you're going to get inflammation, and you're going to induce the induced inflammatory response. So you're going to activate macrophages and dendritic cells and neutrophils. And those are going to move to the site of infection or pathogen exposure and hopefully eliminate the pathogen before it makes it further into the system. This works for most of the time. Most of the time we're exposed to a pathogen, our innate response and our induced inflammatory response does a great job of eliminating it. But every once in a while we need some heavier guns. And that's where the adaptive immune response comes into play. These dendritic cells and macrophages, primarily the dendritic cells, will take the antigen from the periphery and bring it to the lymph node. In the lymph node, B cells and T cells are patrolling and monitoring. So in the lymph node, all that antigen from the periphery is being presented to B cells and T cells. If the B cells and T cells react, they become activated. And they become activated in the lymph node to then produce the CD4 T cells, the cytotoxic CD8 T cells, and antibodies. So in this way, you start from the periphery with your innate response. If that doesn't work, you have induced inflammation by the induced inflammatory response. And if that still doesn't work, you then employ your adaptive immune response to take out the pathogen because it's highly specific and it's more targeted. All right, so before we conclude, the way I'd like to end is to go through a little bit of a thought experiment of how we manipulate the immune system. So to do that, we're going to talk about development of a vaccine. Because if you understand the development of a vaccine, you really thoroughly understand the process by which cells are activated in the body. All right, so the scenario is this. You have Haemophilus influenza B. Haemophilus influenza B is a polysaccharide-based bacteria. And it's really dangerous because it causes epiglottitis in young children. So your task is to make a vaccine against this. Okay? You want to make sure that no more kids get sick with epiglottitis. So the first thing you got to think about is, okay, what do you want from this vaccine? Well, if it's a bacteria, you don't want it to be able to infect the body. So ideally, you want an antibody, because an antibody can coat that bacteria and then it's not a problem, right? So the first thing you want is an antibody. So now we're going to work backwards, and we're going to say, okay, our goal is an antibody. What do we need for an antibody? So the first thing is we need a B cell, because B cells make antibodies. So then we got to work further. We go, okay, so we want B cell activity. What do we need for B cells? Well, we first need a B cell receptor. And because of the nature of B cell receptors, because they can respond to polysaccharides, proteins, and different inorganic material, there is likely going to be a B cell receptor in the body that reacts to that polysaccharide. So thumbs up, B cell receptor exists because that's what our body produces B cells for, all right? So you don't have to worry about making the B cell receptor. It's likely going to exist. All right, so to get that, though, you need the antigen present. So the B cell receptor needs to be exposed to the antigen, all right? So to make this vaccine, you're like, okay, I've got Haemophilus influenza. What am I going to do to it? Well, if you grind it up so that the polysaccharide is present in a little vaccine, and you inject it in somebody, well, that polysaccharide is exposed to the immune system and can be an antigen presented to a B cell. All right? So to get that antigen presentation, you grind up that Haemophilus influenza and you inject it. All right. Turns out if you do that, though, you don't get immunization. It just doesn't work. And if we think about why, we've got to look at our activation pathway and really reason out why did this fail. And what that comes down to is we neglected the fact that B cells need more than just a B cell receptor. They need coactivation. And so then we got to go, okay, well, shoot, what did coactivation require? And coactivation requires CD40 ligand from T cells. All right, so now we got to twiddle our thumbs and go, okay, so now we need T cells active. And what do T cells need? Well, T cells are going to require, that's right, a T cell receptor activation. All right? We got to trace it back. And what does a T cell receptor require? Well, a T cell receptor interacts with MHC. Okay, so we got MHC, and that's all over in the body, so we don't need to make MHC, it just exists. But we have a problem because MHC requires protein. And if we remember, our Haemophilus influenza is a polysaccharide. So when we gave the Haemophilus influenza, it just didn't get attached to an MHC because it's not a protein. So what do? Well, we just add a protein to the mix of our injection. And the question is, what protein do we use? Well, any protein that is inflammatory will work. And the way this is done is you take an inflammatory protein, often this is some sort of a toxin, and you attach it to the polysaccharide. So in a lab, you just attach one to the other. So you give the polysaccharide for the B cell, and because that B cell takes up that polysaccharide with its receptor, it then has access to the protein to then present on MHC class 2. So by attaching a protein to the polysaccharide, you essentially give the protein that is needed for the T cell activity. All right? And that's what we call a conjugate protein, because the protein is conjugated to whatever you're giving as a vaccine. All right? So it turns out you try this. You attach a protein, you put it in a shot, and you give it to some people. And it still doesn't work. You still don't get immunity. So we've got to go back to the drawing board. And we've got to go, okay, what are we missing? Is there anyone who would like to volunteer the step that we have neglected thus far? Right, the co-activation. Right now, you've addressed everything except for T cells require 2 signals, not just one, so you need co-activation. So, does anyone remember what you need for co-activation of a T cell? What? B7. Where does B7 come from? Sorry, louder? I can't hear up here. Where does antigen-presenting cells, macrophages and dendritic cells, right? So it turns out what you need is you need the activation of a macrophage or a dendritic cell. All right, and this is what we call an adjuvant. An adjuvant is going to activate that innate response that you need to activate the T cell, okay? Adjuvants, there's many of them. One of them is alum, but you don't need to know the specifics of what an adjuvant is or what molecules are adjuvants. What you do need to know is that an adjuvant facilitates immunity by activating the innate response, okay? So if we put it together, from the beginning, B cells required T cells required proteins and macrophages, and macrophages require an adjuvant. So in total, what we've done is we've taken our polysaccharide of our capsule, and we've attached a protein to it, and then we've thrown in some inflammatory stuff in with it, and we inject someone and successfully have made a vaccine. All right? The reason I do this is because it really ties together everything we talked about. It ties together the innate response all the way to the adaptive response. And as you move into the clinical immunology, a lot of the clinical scenarios are a matter of regulation and pathways. So if you know what begins a pathway, for example, if you know that neutrophils contain NADPH oxidase, and someone tells you that, oh, you lose NADPH oxidase, well, you lose neutrophil function. So as you're reasoning through the clinical immunology in the coming days and weeks, I would try some of these thought experiments. Try reasoning out why is it that when X happens, Y is what we see as the pathology. Because often with the immune system, you can trace it back in a pathway. And I think that's what's really beautiful about the immune system. You may completely forget the disease and how it presents. But if you know the protein involved or you know where it happens in a pathway, often you can predict how it's going to present. So with that, I would like to say thank you all for coming. I hope that this was helpful. And I wish that I could, thank you. I wish that I could stay to help answer questions. Unfortunately, I have a meeting in Skyline that I have to run to. So Dr. Hamby, Tiwari, and Dr. Galipta are going to be here to help answer any questions that you have. But before I leave, I also want to say thank you so much to the 3 of them for allowing me to come and participate in the immunology course this year. Best of luck to all of you, and feel free to contact me if you have any questions.